Hello everybody. In my last lecture, I talked about the visual signal processing from the eye to the primary visual cortex or V1. We talked a lot about the signal captured by the eye, right? And how the signal is processed in the retina and how the action potential that travels from the retina to the primary visual cortex, which is the visual pathway from the retina to the brain. And you also know that from V1, the signal um, goes to V2 and then V3 and then two different streams or pathways. So as the signal goes to more higher processing areas, the function becomes more complex and more cognitive functions are involved. That means the higher visual processing takes place in the latter areas. So earlier areas like striate and extrastriate areas. Striate area is the V1 and extrastriate V2, V3. In those areas, the earlier visual processing take place, and the areas located in the temporal and parietal lobes. That means the ventral and dorsal pathways. In those areas, higher visual processing take place. That means those areas in the ventral and dorsal streams, those areas are involved in higher processing. Now, which functions are considered as higher visual functions? Not just seeing the image of something which is processed in V1, V2. However, higher visual functions include the visual attention, searching, visual searching, higher motion processing, face and object recognition, visual depth perception. So these are the tasks belong to higher visual functions. So you already know that the primate visual system or visual part of the brain consists of about 30 areas. So a large part of the primate brain as well as the human brain is involved in visual processing. We mentioned that before. And in human brain, there are more than 35 visual cortices or visual areas. In primate, about 30 areas are present in the visual part of the brain. So far, we know there could be more. And two streams I mentioned in my last lecture. One stream is the ventral stream, which is also known as hot stream. That means object recognition, and that hot stream or ventral stream is located in the temporal lobe, inferior temporal lobe. And the dorsal stream, which is also known as wire stream, is located in the parietal lobe. So here you see this is the parietal cortex and this is the temporal cortex, temporal lobe. This is the parietal lobe and this is the dorsal stream or pathway, and this is the ventral or inferior temporal, right? So this is the temporal lobe, and this part is the inferior temporal part. So you have the ventral stream in inferior temporal and dorsal stream in the posterior parietal, because this is the parietal cortex, right? And this part is the posterior part, this is the anterior part, right? So posterior parietal. So those are two pathways. And in this slide you can see different important visual areas in the dorsal and ventral streams. Okay, so one thing is just to remember that many of these dorsal stream areas named after parietal. So you'll see P. So that is one clue that these are the areas in the dorsal stream. So we know that the signal, after the signal arrives in V1, it goes through V2 and then to the dorsomedial area. The first area in dorsal pathway is MT, which is MT is in monkey, and in human homologous area is V5. Okay, and then to the inferior parietal lobe, and in the inferior parietal lobe you have some important areas: LIP, lateral interparietal area; CIPS, caudal interparietal sulcus; VIP, ventral intraparietal. So all those are inside the parietal, and anterior interparietal or AIP, and then superior and inferior parietal lobe. So SPL, superior parietal lobe, and IPL, inferior parietal lobe. So these are the areas, important areas in the dorsal stream. Now if you see the ventral stream, the areas are in the inferior temporal lobe, and in this stream from V1 goes to V2 and then to area V4, and from V4 then it enters into the temporal lobe, inferior temporal lobe. Important areas in the temporal lobe are fusiform face area, FFA, and lateral occipital cortex, LOC, also known as LO, parahippocampal place area, PPA, okay, posterior inferotemporal area. So these are some important areas in the temporal lobe, that means in the ventral stream or heart pathway. Okay, so here you can see in this picture, first the signal arrives in V1 here, right, then V2, and then some signal goes to V3 and V4, and from V4 goes to inferior temporal this way, and this is another lobe this way, and from MT the signal goes to the posterior parietal. Okay, so this is the ventral and this is the dorsal. Now if you see the bottom of the temporal lobe, this is the temporal lobe, you can see a couple of areas here: fusiform face area and parahippocampal place area. Those are side by side in the inferior temporal surface. And fusiform face area is an interesting area in the ventral pathway. The neurons of this area are sensitive to face, so these neurons excite or start to fire when you show the person or monkey the face of someone. So these are face sensitive neurons in the fusiform area. However, parahippocampal area neurons are sensitive to landscape, larger, landscapes like house or, a mountain, those kind of things. So interestingly, two areas are responsible for two types of uh, functions. Okay, how the ventral stream works. Ventral stream works as image libraries. So within the ventral stream, the image libraries are stored. That means the recognition of faces, shapes, objects, and routes is attained by matching what is seen. So the information is already stored there, and when you see, the areas will match with your stored information and will give you the recognition of that object. Okay, like if you see an apple, you have the memory of an apple, how it looks like, right? What's the shape or color? All those information are already stored, and the ventral stream areas have that information already, and when you see it quickly matches and gives you the perception of object recognition. So for object recognition, we use the shape, right? Edges, color, the curvatures. So all those, the size. So all those are processed in the ventral stream. On the other hand, the dorsal stream serves the function of appraising the whole visual scene, because it is a where pathway. So to see the things, where the things are located in your visual field, you need the bigger area. You need to see the bigger area, right? So dorsal pathway works with the large area or whole visual scene. Okay, and the dorsal pathway works along with the frontal lobes, because frontal lobes are very much related to the attention. So when you see something is moving from here to here, the motion, you need to follow it, right? So you have to pay attention that from where it is moving to where. You need a continuous attention to watch that object motion, how fast, how slow, if it is getting fast or slow. So all those information you need to up to date. So that's why the frontal lobes are more involved with the dorsal stream functions. And dorsal stream facilitates immediate visual guidance that I just mentioned here of movement through the scene. And by interacting with area V5, which is MT in monkey, this area is highly sensitive to direction and speed, that means motion. And I worked on this area a lot on monkeys. Okay, and it is not memory based, right? Like when you see something is moving, you don't know where it is heading to. So it is is not dependent on the memory. You just immediately recognize by looking at it. Here in this picture you can see a few areas. These areas I have already mentioned, dorsal stem or higher areas. Here you see three different areas: AIP, LIP and MIP, and three areas have three different types of functions. That means the neurons are sensitive to three different types of functions, are involved in three different types of functions. MIP, the neurons in this area are responsible for or involved in planning visually guided reach movement. So when I follow something is moving, like a baseball, the player already have hit the ball, so the ball is now approaching towards you. You are keep watching, right, the movement of the ball, and also you move your hand to catch the ball. So that is the function of MIP, involved in planning visually guided reach movement. Okay, or if I give you a cup of water, put on the table, you have to look at it and measure the distance, the direction, right, shape, all those information, then you move your hand. So visually guided, you have to see to grab it. So when you do that movement, the MIP neurons get activated. So by looking at that, we can tell that MIP neurons are involved in that task. Not LIP or AIP neurons are activated in that situation. However, LIP neurons get activated or involved in the control of eye movement, including intended eye movement. So when you move your eye, like if I ask you that look on the computer screen and a green dot will appear anywhere, okay, you don't know. Now if I tell you that the dot will appear in top upper right corner, so now you have the plan, right, that you have to move your eye towards that direction. That is the intended eye movement, because you have the plan, and when you move your eyes, only then the LIP neurons show huge firing or activation. Okay, so we can tell that this area or the neurons of this area are involved in intended eye movement. Now AIP is involved in visually guided grasping movement. Okay, so after you move your hand, then final action is grasping. So for grasping, you need more information, like to reach towards the cup, you don't need the information about the shape of the cup or the handle where you will hold it. You don't need those information. You just need the information about the distance and direction, right? But when you grasp it, you need more information about the shape and the orientation, all those things, right? So AIP neurons get activated during this final action, grasping movement. Okay, so you see that these three areas are involved in three types of functions, although those functions are related to each other, right? Because you have to move your eye, intended eye movement, you have to reach the object, and finally you have to grasp the object. So all these areas are involved, but they are involved differently, performing different functions to accomplish the complete function. Make sense? So therefore we have many different areas in the visual part of the brain, and they are processing different functions, but they are coordinating or talking to each other or working together. And all these functions are where function, right? So you need to know where. Lesion study gave us a lot of information about the function of these areas. So many studies were performed by lesioning the areas, and mainly on the monkey's brain, because monkey's brain is close to the human brain, you know that. So there are similar areas in the monkey's brain. So we use the monkey to study the functions of those visual areas. Okay, now you know that if we make lesion in the dorsal pathway or damage in the dorsal pathway, that deficits in visually guided reaching, okay, which is the damage in the posterior parietal cortex. Make sense? Where pathway. Now ventral or hot pathway damage or lesion causes the monkeys severe difficulties in visual recognition or object recognition and discrimination learning tasks. Make sense? So by lesioning different areas, we can test the functions of those areas, and that's how we know that dorsal areas or dorsal stream areas are involved in where function, and ventral areas or ventral stream areas are involved in hot functions. How the experiments are done. This is a nice example of the experiment on where and hot pathways. Very simple experiment on the monkeys, and you can get an idea. So this experiment was designed this way. You see that in this case, the first one, this is a landmark task. That means where task. So the food was placed under or hidden under a square rectangular bin. Okay, so this is the shape of the bin, and under this rectangular bin you have the food. So monkeys were trained on that. So monkeys now know that the food is the cue for the food is this rectangular bin. Make sense? So now here in where task, what the experimenters did, you see here both these two bins are rectangular. So monkey doesn't know which one has the food. One of these two has the food. So in this case, you give a cue to tell the monkey in which bin the food is located. Make sense? So this is a where task. So this is the cue, this structure. Now when I put it here close to this bin, monkey knows that food is in this rectangular bin. If I move this one from here to here, put it here, then monkey knows that the food is in this bin, right? So this is a task involved the higher or dorsal stream. Make sense? So monkeys are locating the position of this cue, and based on that it is finding the food. And now if you cause the lesion in dorsal parietal area here, which is the higher stream, then the monkey cannot recognize or cannot detect the food. When before lesion, you place every time you move the bin, you move the cue from here to here, the monkey can correctly locate the food, like 100% accuracy. Okay, so wherever you put the cue, the monkey knows that the food is in that bin. However, after you cause the lesion, 50% cases the monkey will do the mistake and 50% will do the correct. That means half, which is like random. The monkey cannot locate the food. That means the dorsal parietal areas are involved in higher task, right? Because the higher task is lost after the lesion. Now, this is another experimental design. In this design, the monkey has to simply just decide where the food by looking at the shape, recognizing the shape of the bin. So as I told you, that monkey knows that rectangular bin has the food, right? So the monkey has to do what? Only just recognize if it is triangular, and by color if it is red or green, rectangular. So two things the monkey has to just detect: the shape and the color. That means what the object is. And every time you move the rectangular bin from one position to another, the monkey would correctly recognize the rectangular bin to find the food. However, 100% accuracy. Now, if you cause the lesion in the inferior temporal area, then what happens? 50% cases the monkey will say that food is here, will look for the food here, and 50% here. That means chance level. So actually monkey cannot recognize the shape or color of the object. Makes sense? So this is a simple experimental design, and but it clearly tells you that which areas are involved in which functions. In human, lesion in the dorsal stream. In human, we cannot make the lesion, but lesion can occur due to injury or external injury or internal cerebrovascular accident, like clot or stroke related damage. If we see that in human brain damage or lesion occurs in the dorsal stream, in that case the subject is unable to reach out and grasp the object. That means it cannot really perform the visuomotor reaching and grasping task, which are the higher tasks. We talked about that, right? So that clinical condition is called optic ataxia, and the problem is not purely visual and neither purely motor. It is a problem in visually guided motor action, right? So this visually guided motor action is involved in this lesion. That means the person fails to perform visually guided motor action. Okay, so that is skill of motor action is lost. The patient in optic ataxia, this is the pseudonym of that patient, okay, can verbally describe the orientation of an object. Like if you show the object and tell you if it is this way or this way. Okay, the subject, by looking at the object, the subject can correctly tell that yes, this is, anteroposteriorly, this is superiorly, whatever the position is. Now, if you ask the subject to hold it, then the subject cannot do it correctly. If you ask the subject that every time you will move the object, put it in different directions, and every time the subject has to hold this way, like one finger here, another finger here. Okay, now the subject cannot do it correctly. Okay, so that is the visually guided reaching and grasping deficit, movement deficit. Makes sense? So let's see what happens here. You see the subject was asked to hold this object this way, and sometimes the subject was asked to hold it this way. So the subject got the instruction how to hold the object, and this is the normal subject, okay, control. So you see that normal subject definitely hold the object correctly, holds the object correctly, right? So these are the directions the subject were asked to hold the object. So all these are correct. Okay. Now this is RV, the lesioned subject, the patient, and you see when same instruction was given to RV, he failed to hold the object in right direction or correct direction. So very different than this one. This is normal, correct tasks, and here you see by the lines you see the orientation of the fingers. Same thing here, another subject, and normal condition and RV's response. So the RV patient fails to hold the object in right orientation or reach and grasp the object in right orientation. However, another patient, DF, whose ventral stream was lesioned or damaged, and this person, can you see, hold the object correctly, which is very similar. Now if you compare, this is control, this is DF, you see the same direction was given and these two are very similar, right? These two are very similar, these two are very similar. So the DF patient did not have that deficit of reaching and grasping the object, visually guided reaching movement. However, RV had that, but DF, since DF had lesion in ventral stream, he had the problem which are different than higher task. he had a problem in hot tasks. So you see here, visual motor task was intact that we saw before. However, DF cannot identify by looking a face. So face recognition was lost. You must remember I mentioned fusiform face area in the ventral pathway, inferior temporal, and the lesion causes that problem. The subject cannot recognize the face, but can identify the person by hearing the voice. Make sense? Or by name. So the patient can identify by hearing the voice of that person or hearing the name of that person, but cannot identify by looking at the face of that person until the subject, the person talks. The subject talks, the patient cannot identify. So this is purely, deficits in recognizing the face. Unable to copy line drawing, but can draw the object reasonably well from long-term memory. So this is another interesting observation. If you show an apple and ask the subject to draw exactly, what the subject sees here, you see how DF, sketched this apple, which doesn't make any sense. However, if DF was not shown any apple but was told to draw an apple from his memory, he can do it in some extent. That means by looking at something, its shape or size or curvature, it cannot follow the object, but from memory where it's already stored, he can do it, right? So instant object recognition is lost. When the subject was asked to draw a boat by looking at the boat, he cannot do it. But when he was not shown the boat, but he was told that draw a boat, he can do in some extent. So that is the condition called visual agnosia related to ventral stream lesion. Okay, now we'll talk about an area which is heavily involved in motion recognition. That means two vectors of motion are, I already mentioned, direction and speed, right? So these neurons of area MT in human V5 are heavily involved in motion recognition. This person, William Newsom, he was actually the mentor of one of my mentor, D. Angelis, who used to work at WashU, and I worked with him. He first, identified that area MT is a chunk in the deeper part of the occipital lobe where you have discrete neurons sensitive to motion. Okay, so he performed many experiments and proved that this area is sensitive to motion. So let's see here. This is the tuning curve of an MT neuron. So one single MT neuron, you put the electrode and record the activity of this neuron, MT neuron, and when you do it, you present the dots. For example, on the computer screen, this is the computer screen, you put the dots, many dots, and the dots are moving to different directions. So randomly you change the direction, computer program changes the direction, so the dots move, and you keep recording from this neuron. And at one point you see the neuron starts to fire heavily, strongly, when you present the dots, move the dots to a particular direction. For example, this neuron fires like this when you move the dots, or when the dots move rightwards. But when the dots move to other directions, this neuron is quiet. Okay, just a spontaneous firing. Okay, or even inhibited. So this is the preferred direction, right, of this neuron. Another neuron might have preferred direction this way, another might have preferred direction this way, right? So this is the tuning curve of one neuron, MT neuron, and this is the preferred direction. That means I'll say that 100 probably around, like in between 90 and 110, that's here 125 degree. So this is the best or preferred, most preferred direction of that neuron. Okay, another neuron might have the tuning curve like this, another might have the tuning curve like this. So in MT you have the neurons prefer different directions, okay, and specific directions, not other directions. It has broad tuning curve in this neuron, so it responds in some extent when the dots move to 90 degree or 170 degree too. Some cases we see very narrow tuning. That means this neuron is very selective to a particular direction. So some neurons have narrow tuning curve, some neurons have broad tuning curve, but clearly the neurons have preferred directions. Okay, and now this is the histogram showing that MT neurons respond to a particular direction, and this is from another neuron, and spikes per trial. So you see the firing. If you go this way, the firing or action potential increases. Okay, that means the neuron fires more this way and less this way. Okay, so this neuron, the motion coherence for this neuron is 12.8%. What is motion coherence? Let me just explain that first. Like I am presenting the motion stimulus on the screen, and these are the dots. Okay, so rastergram. These are the dots. So you see, if all these dots move together to same direction, that means 100% coherence. Makes sense? If half of the dots move to one direction, same direction, and other 50% dots move randomly to many other directions, that means 50% coherence, right? So here the dots were presented at 12.8% coherence. That means about 13% neurons. That means 13 out of 100 dots, not neurons, moved to one particular direction. That was strong enough to stimulate the neuron, MT neuron. That means the MT neuron is very sensitive to direction. Makes sense? Even when 13% dots are moving towards one direction, preferred direction, the neuron increases the firing a lot. You see the firing rate has increased significantly. So the blue bars are showing the preferred direction, and the red bars are showing the opposite direction. Okay, so motion in opposite direction. However, same neuron, if you lower the motion coherence, that means 0.8% in this case, in this experiment, that means less than one dot moves towards the preferred direction, one direction, and others are moving all different directions. So this is a very extremely weak motion stimulus. Makes sense? This one is somewhat strong, but this is very weak. It doesn't have any information, motion information, because the dots are moving all different directions randomly. So scrambled, no actual motion is seen here. But here 13% dots are moving towards the preferred direction and others are in opposite directions. Some motion signal is here, but no motion signal is here. So in this case, even if you move that 0.8% dots towards the preferred direction, you see the firing is very low, close to the non-preferred direction or opposite direction shown by red. So red and blue, no difference. So no motion signal is here. That's why here you have some motion signal. But of course, if 100% motion coherence is present, that is extremely strong motion stimulus. Makes sense? Even 50% will be strong for this neuron, because you see only 12.8% gives a very strong, increase the activity of the neuron, MT neuron. So this experiment clearly tells that MT neurons are very sensitive to motion. This is how we used to present the motion stimulus on the computer screen and at different coherences. Here you see 100%. Makes sense? Because all dots are moving together in the same direction, 100% dots. And here 33%, that means 33% dots are moving towards the preferred direction shown by yellow, and others are like moving randomly. So this is much weaker than this one, but for MT this 33% is even very strong. Okay, so that's how we perform the experiment and present the stimulus, motion stimulus on the computer screen. So monkey has to look at the dots and we record the activity of the MT neurons. Now here you see this is a nice experiment. The recording from V1 neuron and MT neuron, and you see the comparison. Okay, so in three conditions. First, you of course put the electrode and record the V1 neuron activity of the neuron, and now you present the stimulus. Okay, so the stimulus was presented in three conditions. Blank screen, so monkey, this is actually a human experiment. So we no, this is a monkey experiment, so recorded from area MT, but this is fMRI. So you just see the activation of the area. So three conditions: blank screen, moving dots on the screen, and stationary dots. Okay, and you see when the screen was blank, it is like, no signal is there, so no activity here. Makes sense, right? No activity here, blank, this color. Now when you put the moving dots on the screen, you see V1 neuron increased the firing a lot, right? Signal change, increase the firing a lot. Now again blank, no firing, baseline. Now you are presenting stationary dots, not motion, but stationary. You see, same way, the neuron increased the firing a lot. So no difference between this one and this one. That means no difference of response in stationary dots and motion dots. That means dots are stimulating the V1 neuron, not the motion. Make sense? However, when you record from MT, you see a different picture. Blank, no response, makes sense. Motion dot, huge increase of firing. Blank, no. Now you are presenting stationary dots. Again, here, no firing, no increase of firing. So the difference here, this one, V1, are sensitive to dots. It doesn't care whether it is motion or non-motion or stationary. In both cases, it will fire when you show the dots. But MT only fires when the dots move. So MT neurons are sensitive to motion, not stationary object. Now this is another interesting experiment on MT. You see on the screen two sets of dots, black dots and white dots. Black dots are stationary and white dots are motion dots. Okay, and you record, this is a human recording from the human brain, fMRI experiment, and the experimental design is pretty simple. Each set of dot will appear at a time. So when the black dots, stationary dots will appear, both sets of dots will be presented at the same time, but the subject, the human subject, was asked to shift the attention and was instructed verbally. So the experimenter will tell the subject, pay your attention on dark dots. So the subject will pay attention on the dark dots. Okay, and then you are recording fMRI, and then the subject was asked to shift the attention from dark dots to the white dots. White dots are in motion. So the subject will just shift the attention from the black dots to the white motion dots. And then again you ask the subject, move your attention to the black dots. Okay, that way, alternating. And here you see the activation of area MT. So when the subject was asked to pay the attention on the white dots, remember the subject is seeing both dots because they are presented together. So they are the subject is seeing both, but just changing the attention from white to black, black to white. When the subject pays attention to the white dots, that means the motion dots, you see the significant increase of the firing here. But when the subject was asked to shift the attention to the black dots, the firing is very low. That means spontaneous, right? So That again tells you that one thing, number one, the MT neurons are sensitive to motion, but not only motion, but the attention that is being paid to the motion. So attention related motion sensitivity. Make sense? Higher order visual function, because attention is also, it is not simple motion but attention related motion. So that's how the experiments are done. This is another experiment. In this case, the recording was performed on V3, area V3A. Okay, and interestingly, V3A, area V3A is sensitive to the actual motion of an object in the environment, not the apparent motion, not the motion of the retina, the actual motion of the object. Now, two different situations. Let me explain. Like here, this is your eye. Okay, your retina is here, and this is the object, and this object is moving from this position A to position B. Okay, here to here. Now your eyes are stationary, not moving. So when the object is moving from here to here, A to B, the picture is moving from one location to another. Retina is fixed because eyes are not moving, right? But the picture or image is moving on that fixed retina. So that gives you the perception of actual motion. Make sense? Now another condition here. You see, this is your eye and your retina is here. Now the object is fixed here, not moving. If I ask you to move your head, okay, move your head, that is moving the picture of the object on the retina, because you are now moving the retina, object is fixed. In this case, retina is fixed but object is moving. That's the actual motion, right? And in this case, the object is fixed but the retina, you are moving the retina because you are moving the eye, you are moving the head. Okay, so the B3A neurons are sensitive to the actual motion, not this. Okay, so that is the experiment. So here now you will understand, this is the monkey sitting, head is fixed. Okay, so the monkey was asked to fixate eyes here. This is the fixation point. Okay, and the monkey will not move the eye, must maintain the fixation. So eyes are fixed, not moving. Now you on the computer screen or real object, whatever you do, the object is moving from this position to this position that I mentioned in this case, similar situation. So the object is moving from here to here, and when the object is moving over the receptive field, this is the receptive field of that neuron. So you are recording from a neuron of V3A. So this is the neuron of V3A, and this is the electrode. You already placed the electrode and got the neuron. You are recording now. Okay, now when first thing you do, you will find the receptive field of this neuron. So this is the receptive field here of that neuron. Okay, so when you move the object and the object passes through the receptive field, you see huge increase of the firing of V3A neuron. Okay, now this is a condition similar to this. What you are asking the monkey to do is, you are not moving the object. The object here, the red bar, is fixed here. Okay, and this is the receptive field, same neuron, same receptive field, right? Same location. Now what you are asking the subject, monkey, to move the eyes from this fixation to this. So when the eyes are moving, this object, the receptive field is also moving. So receptive field is moving like this, moving this way. Make sense? So the object is passing through the receptive field, or we can say the receptive field is passing through the object, although the object is fixed, right? So retina is moving in this condition. You don't see that huge increase of firing, slight increase, but you don't see. So we can tell that area V3A neurons are sensitive to actual motion or movement of the object. So you see that different areas have different interesting functions, right? To process different types of visual signal. Okay. The higher order motion processing includes the motion in 3D space, motion parallax, optic flow, and inertial motion processing. So let's see the motion parallax. What is motion parallax? You all know that when you go to a ride, the virtual ride in like Disney World or Disneyland or somewhere, you are sitting on a chair, or you are sitting on a bike, and in front of you huge screen where you see the dots are moving, and although you are not moving, you are sitting on the chair or the bike or in a stationary car, but the dots are moving, and that is giving you the perception of movement or motion, right? It's pretty strong, that those rides are for creating the optic flow. So this is not an actual motion or real motion. This is just the motion created by the dots, and this type of motion is called the optic flow. Okay, generated by optic flow. And when the dots on the screen in front of you, huge screen, moves one way, you feel that your body is moving to the opposite direction, and that is the optic flow. Okay, so now just think about the real situation. We are experiencing the optic flow in real world all the time when we are moving. Makes sense? Because everything in front of you, just think that everything is representing the dots, and when you move forward, expansion occurs. The things are expanding. When you move backwards, the convergence or squeezing of the field takes place. So when you move forward, the things you are looking at in front of you in your visual field are expanding. Okay, you see here expansion. And when you move backward, everything is squeezing or convergence. When you turn rightwards, the things in front of you move leftwards. When you turn leftwards, the optic flow occurs rightwards. Okay, so just opposite. So that is the real situation. Now, if you are stationary, I show you, I move the dots or the things in front of you, then you feel the motion in opposite direction. Make sense? So that is the optic flow. And for optic flow, you need a huge field. Within a narrow aperture, you cannot feel the optic flow, right? And there are two important areas. Both are sensitive to motion. One I have mentioned is the MT. However, next to MT, very close to MT, there is an area that is called MST. MT is middle temporal and MST is middle superior temporal. And the main difference between MT and MST is MT neurons are sensitive to the real motion in small visual field. Like if you see an ant is moving, okay, so that motion is processed in MT, in a smaller area. However, MST neurons are usually sensitive to the motion in larger field, bigger field, like optic flow. So for optic flow, you need to process the optic flow, you need the area MST. MT cannot do that. Okay. Also, for the motion from the vestibular signal, you must remember semicircular canals and vestibule. So vestibule in your ear, the movement of the head or body gives you the information about the motion, right? And MST also processes the vestibular motion signal. Okay, so optic flow, vestibular motion, those signals are processed not in MT. So far we understand in the MST. Okay, and those are not real motion. The vestibular is real, but visual optic flow is not the real motion. Now, MST is divided into two areas: MST-D, dorsal, and L, lateral. Okay, so MST lateral, MST dorsal, and these two areas work slightly differently. MST lateral senses real object motion, like a flying bird, and smaller than MST-D, the visual field. Of course, MT is much smaller than MST-L, but if you compare these two MST areas, MST-L lateral works in a rather smaller field than MST-D. Okay. MST-D dorsal senses the visual motion produced by the self-motion, optic flow, and vestibular signal processing. Okay, but if you compare both with MT receptive field is much smaller than MST-L and MST-D, of course. So here you see these two areas, the location, just in our rough location, MT, the yellow, and MST, the red, and MST is divided into dorsal and lateral, right? This is actually the recording of the receptive field. I recorded the neurons of MT and MST, and you can see from my recording from monkeys area MT and MST, and you see the comparison. This is the receptive field in MST neuron, and this is the receptive field in MT. So in MT, the receptive field is very small compared to MST. Okay. And I also found that many MST neurons are very sensitive to the motion of the body, that means the vestibular signal. But MT neurons are not sensitive at all to vestibular motion signal. Okay. And also MST are sensitive to optic flow. I recorded many neurons are very sensitive to optic flow. Large screen I used to create the optic flow and recorded the MST activity and found that many neurons are very strongly sensitive to optic flow and larger screen, but MT neurons are not. But when you present the motion in a small aperture, the MT neurons are sensitive to motion. Okay. So those are the things you need to know in this part of my lecture. So I'll stop here, everybody.